Hi, everyone. Thank you so much for joining us today. Before we kick this off, I do want to point out to the audience that we welcome questions. Please post them in the Q&A section at the bottom of your screen, and we will address those questions at the end. So I'd now like to turn this over to Stephen Saltzstein, Force Family Office's CEO.
Thank you, Callie, and thank you everyone for joining us today. We greatly appreciate you taking time out of your day to be with us, especially after the close here. We have a fascinating, very timely discussion with Robert Bitterman, CEO of Phio, and Jennifer Phillips about skin cancer and what Phio is doing in order to create drugs that can treat skin cancer. So, Bob, you have a wealth of experience and a really strong background. I guess my first question is, how did you end up in RNA science?
Okay, but that's a fair question, Steve. So actually, I was asked to be chairman of the board of what was Phio before it became Phio. It was called RXI Pharma. And it was basically a small molecule dermatology company. It was developing drugs to treat other types of skin conditions. And I was asked by the CEO if I would, who I had known for a number of years, if I would be willing to volunteer my time in basically a passive governance role in order to oversee what was going on, but mostly governance, right? And so that's where I got involved initially. And then the company basically pivoted into immuno-oncology around 2017, 2018. And honestly, my knowledge of immuno-oncology was not that much. And from there, as some of the changes in the organization took place, I was approached as we had to replace the CEO sometime around 2022. And we were seeking recruitment, and we were not finding the candidates we were looking for. And eventually I was asked if I would be executive chair to take on a bit of an operating role, but still not taking over as president and CEO. Sometime around the beginning of 2023, at that point, the board came to me and said, "How would you like to take on the whole thing?" And I thought about it for a little bit. I had become pretty much immersed in what the technology was. And honestly, I was in a stage of semi-retirement at the time. And I said, you know what? I'll do this if I can find some other colleagues of mine from my past life that were excellent at drug development, and if I could talk them into coming along with me. And so I talked Jennifer, who's with us here today, and another of my senior colleagues, and they both came out of retirement. And we took this thing on because the science was fascinating. It had an opportunity to make really medical impact, and we thought the opportunity for return on investors was going to be fantastic. So with that, we then surrounded ourselves with a handful of other people who we'd worked with in the past for a number of years who were experts in drug development. And that's the key, because we had to transition from the company from a research discovery company, which is very different than going in and actually doing drug development itself. It's a different game. But that's how I got here.
Okay, great. And just in simple terms, I mean, what is the company trying to do?
Ultimately, with this technology and understanding the condition that we're treating, skin cancer and particularly squamous cell carcinoma, what we're trying to do is fill an unmet patient need that exists today. And with this product, this technology, turning it into a drug, guiding it through the regulatory development pathway to a point where it can be commercialized. That's the ultimate goal. It's a challenging one, but right now I believe we have the right people with the right structure to pull that off.
And so the lead program targets skin cancer. Can you just talk to us, specifically about that, and maybe a little bit about the minor players in the rest of the pipeline?
Sure. So skin cancer is in the medical world, they call it cutaneous carcinomas, okay? But we'll stick with skin cancers. And there's a few different varieties of them, the largest of which is basal cell carcinoma. And many people who've been exposed to sun excess UV radiation over their life, they end up with basal cells. They're typically removed by surgery, by a derm surgeon. Sometimes there's less invasive approaches. That represents the largest segment of this. There's probably about 3.5 million incidents of that a year. The second largest incidence is cutaneous squamous cell, which is where most of our focus is. And then the next one that comes up is obviously melanoma, which is what most people have heard about. That is probably, in the minds of most, the most fatal, if not caught early. And then we have something else called Merkel cell carcinoma. And Merkel cell is something that affected Jimmy Buffett. There was, I think, a recent baseball player, I think for the Chicago Cubs, who recently passed from that. But it's not that common, but it is actually even more deadly because once it starts, it's almost impossible to reverse. Those are the four big pieces. From our perspective, when we basically made the decision to start studying skin cancers back in sometime in late 2022, early 23, we were looking at late stage Merkel cell, melanoma, and cutaneous squamous cell. And when we basically filed our investigational new drug application with FDA sometime in the second quarter of 23, the FDA came back and granted us the opportunity to study late stage Merkel cell, late stage melanoma, late stage cutaneous squamous cell, and also early stages cutaneous squamous cell, stages 1 and 2. And that was important because for us, that was a significant part of the overall market. Now, I don't know if I mentioned this, but cutaneous squamous cell represents about 1.8 million incidents a year. And that is the second largest incidence of solid tumors in the human body, next to basal cell. But it's actually, if you take basal cell out of the equation, because it kind of distorts the numbers, cutaneous squamous cell is more than all the other solid tumors combined in all the other organs of the body. So it's huge. But the stages 1 and 2 are important because they represent about 70% of all the squamous cells. And you might say, well, maybe it's not that important to be treating stages 1 and 2. How bad can it be? Well, the reality is that they can easily and quickly develop into stages 3 and 4. And now that we know that the mortality incidence, as you start moving into the advanced and metastatic cases of these diseases are deadly, it's important that you start treating them early. And right now, the only opportunity to treat them with an approved drug, actually, there is no approved drug at the moment. The standard of care is a surgical intervention. And if you know anybody that's ever gone through derm surgery and they're walking around with bandages on their face, it's not necessarily acceptable for many people. And also, and sometimes it's not appropriate medically because depending on the size of the lesion or the placement on the body, surgery may not be an option. And so what we're doing with this particular program is finding an alternative option for those patients, that either have surgical fatigue because maybe they've been surged many times. And the question becomes, how big of a hole do you want cut in your skin and how often? So it's an alternative. And in some cases, it's just not appropriate. So that's where we are today with the bulk of our work in the study we've done.
Can you talk about the mechanisms of action? Like, how does your drug work theoretically anyway at this early stage?
Yeah. It's an interesting mechanism. It's referred to as short interfering RNA. And let me try to explain this. I'm not a chemist. I'm not a scientist. I'm not a medical doctor.
I'm sure you do a great job.
I'm going to give you the layman's answer to how this works.
Great.
The body, there's a complexity in the immune system. And our DNA is continuously instructing our genes or the RNA to basically deliver messages to do things in our body. And they oftentimes are instructing the body to produce certain proteins. And these proteins, normally in a healthy body, are in balance and everything is fine. But certain proteins in the presence of certain types of tumors will basically cause the body's immune system in the form of T cells which are a primary defense against foreign invaders like a tumor, the presence of the protein may cause the T cells to totally shut down. They become inert, and they are not able to recognize or go after the tumor. When that happens, obviously the tumor proliferates, right? So what our approach is with the technology, we are able to basically look at that signal coming from the RNA that's telling the body to make that protein, and we say, don't do it. We turn it off. We turn that signal off temporarily.
This is what they refer to when they talk about gene silencing.
Exactly. This is what we're doing. That's what we're silencing the message, and we're doing it in a very selective and a very precise fashion because the technology allows us to only target that particular gene that we want to target and not to affect all of the other genes that are doing other things that we want them to continue to do. And so by turning off that signal, it gives the T cells a chance to catch a breather. They reinvigorate, and then they go about their normal business. And the other thing that makes this interesting is that it's different than, say, the approach of what are called the monoclonal antibodies. I don't know if you want to get into that subject or not, but the method and the mechanisms are very different, and I think they're important. For us and for INTASYL, which is the name of our technology, we are getting inside the T cell where that message is being delivered to turn off the protein. So it's done inside the T cell. It turns it off like a faucet would turn off, or in a sink, a faucet turning off the water. So the protein is usually going to be a problem when it escapes to the tumor cell from the T cell. So we're kind of inhibiting that protein to ever get to the tumor cell. In contrast, the monoclonal antibodies, which by the way are infused systemically through the entire body, okay, they are looking to attack that protein after it is already on the surface of the tumor. And so they're going after it's already in place. We're turning it off before it gets there. The problem with on monoclonal antibodies, which are biologic, as they're infused throughout the entire body, they create a number of serious adverse events in the form of autoimmune responses because a lot of systems in the body are being shut down that don't need to be shut down. We're only selectively turning off that which we believe we have to turn off.
You guys recently reported some early patient data. Can you talk about that? And I mean, anything in that data surprise you?
I would not use the word surprise because I don't think we were totally surprised about the mechanism and what it could do. I think we were impressed with the magnitude of what we saw. Because when we chose to work in this area, I'll be honest with you, we were looking at, why did we choose to get into this? Well, there were a few reasons. One, we knew that the market size was huge. Okay, that was important. We felt that it was unsatisfied. But I think the other factor that came into play on this is that we wanted to mitigate risk. And, as a small company with limited funds, whatever we took on as a venture, we didn't want to get a false start. And so we picked a condition which the monoclonals had already validated as being a factor in skin cancer. They had proven this with their monoclonal antibodies. We just felt that we had a better approach with this. And so in doing that, we I can't say that we were surprised, but we were actually impressed with the overall response rate of the patients that were treated in terms of their pathological response. And also we were surprised, not surprised, not the right word, but we were taken back by the level of total clearance among the patients that were treated too. The complete clearance of the cancer. So that was quite pleasant to discover. We expected we would get some response, but not to the extent that we did.
Bob, we also had a very good safety profile with our product as well, which is something worth mentioning here. We didn't have any dose-limiting toxicities or any severe AEs, and this is really important for this patient population who has cutaneous squamous cell carcinomas, because most of these individuals are over 60 years old, so they do have other disease or different conditions that could affect them.
Right.
Yeah. And further to that point, this was a dose escalation study too. So there were 5 cohorts in this study. And
How
with
many
each,
patients?
in total, there were 22 that were treated. But with each passing cohort, the amount of the dose became practically doubled to the point where by the time we got to the final cohort, we were actually dosing a concentration that was about 20-fold higher than the original dose. And within that, we didn't find any dose-limiting toxicity. So the good news about that is that in the 5th cohort, that is where we had the best response rate pathologically. There was an 85% response rate in that cohort. And 4 of the 6 patients who responded actually had total clearance. So that gave us an awful lot of optimism in terms of looking at this and saying we can go with the highest concentration of active, and it is not any different than the lowest concentration in terms of safety, but certainly in terms of efficacy, it was.
So I don't know how you characterized it. Let's just call it a pleasant surprise. So you had a pleasant surprise from
Okay, sure.
from the initial study. Do your scientists or your PhDs like have any theory as to why it had such a strong positive response?
Um, I think they give you a very good reason as to why it was safe. And that was because it was basically just targeting the specific tumor because it's being directly injected as opposed to being systemically infused. And in addition to being localized and being programmed, part of the technology, Steve, is that uh, there's the series of what we'll call fragments of RNA synthetically made that are designed in such a way that they only go after the specific target and they don't
Right.
basically lure themselves away to any other target, right? But there's one other factor in this that's important too, and that the formulation itself is really the drug is delivered to its destination in sterile buffered saline solution. Now, what's interesting is that very few drugs I know can get across the cell membrane of a tumor or any cell membrane for that matter without the aid of all sorts of delivery enhancements. And there are a few of those that are used quite frequently. Lipid nanoparticles are one, also viral vectors. Those elements in a formulation are also known to have toxicities. So we don't have those toxicities either. I mean, this is a really clean way of delivering the drug and doing it through the process of what they call endocytosis. The drug is basically sucked in to where it's got to go. Some people might call it magic, but actually it really is very sophisticated science. But as to why it works as effectively as it does, maybe it's because the drug is basically attacking the source of the problem at its source so that it is not getting the ability to migrate out of the T cell and into the tumor, where the monoclonals then have to kind of break it apart. And now, one of the things we're targeting
I
with our
thought I could be wrong. I thought your drug was injected directly into the tumor. Is that incorrect?
it is.
It
Yes.
is.
Oh,
Yeah.
okay. All right.
So, but within the tumor is the T cell, right? And so that's where it goes, as
Gotcha.
opposed to just being lingering around someplace else in the tumor. It's what's
Gotcha.
known as
Okay.
the tumor. They call it the tumor microenvironment.
Right.
But that is probably the factor that, as I mentioned before, we turn this thing off like water at a faucet. So we don't have to break it apart after it gets connected to something else, the protein that actually creates the problem. So that may be one of the reasons. And if we look at the efficacy rates or the clearance rates of the monoclonals, now granted they're treating a much more serious advanced form of the disease, but where we're getting an 85% response rate overall response rate in that last cohort. And again, it's stage 1 and 2. But the monoclonals, when for the same condition in later stages, their response rates overall are somewhere between like 10 and 17%. So the rates are different, and it may be just the mechanism of action and the fact that it goes to where it needs to go and doesn't go someplace else.
What are the upcoming milestones? A lot of investors want to know, like, what do the next 12 to 24 months look like?
Yeah.
Well,
Well, you want to comment
Stephen,
on that, Jennifer? Go ahead.
sure. Bob has me very busy right now working towards an FDA meeting where we're going to be discussing with them the clinical data from this phase 1b study. And what we are expecting to get out of this meeting is guidance from FDA about the design of our next follow-on study, which we would like to be a phase 2b/3 study, which means that your phase 2 will be incorporated into the phase 3, and this should help expedite the conduct of this study and moving towards submission of an NDA.
That's great. That's great. There are already PD-1s on the market, like Keytruda, et cetera, and they're generating billions of dollars. Do you think your approach is better?
Yes, I do. I think that
Um,
there is a great patient unmet need uh, for a product like this that is very safe, that can be injected in a doctor's office. It's convenient. The physician doesn't need to buy any extra equipment. They don't need an infusion center where you have to make appointments to get into to have that therapy. So I think there's a lot of advantages to our product. And I'm speaking as a pharmacist now. Because I do know that it is very difficult to make these appointments, go and have an infusion, and it's over several months for that type of product. For our product, we have 4 injections. You can go right to your dermatologist office, and you're done within a 4-week period.
Let me ask a question I think everyone's thinking, which is, you had this early clinical data. It looks so promising. Why is the company only valued at like 12 million bucks?
I'll let you answer that, Bob.
Okay. Well, I'm going to let you know. I think today it's 14 million, but you're
Okay. All right. A million here, a million there. Soon it's real money.
That's right. That's right. Thank you, Everett Dirksen, from the old senator from Illinois. But having said that, it's a fair question, and it's easy to give the same reasons that many biotechs will give you, which is, well, the sector is difficult. There's high risk. Lead times are, somewhat long to get to approval. But let's put that aside for a minute, because those are things that we have to deal with. And in our case, we can also talk about the fact that some people believe that there's a significant overhang because of the number of warrants that are sitting out there. But let me address it this way. I think this disconnect between what the science is telling us and where the valuation is today is really the opportunity for investment to actually get a relatively fast return on investment in the near term. Because what we're doing is aggressively today and over the last month or so, trying to close that gap between what the science is telling us and what the investment world and the valuation is reflecting. And one example is, participating in this exercise today with you. We are also very active with several digital companies that are clearly expanding the awareness of who Phio is, what this technology is about, the importance of the disease, the importance of creating awareness. We're busily working on that. Jennifer serves not only as head of regulatory affairs, but also corporate affairs. And so she's basically playing ambidextrous here in terms of both of those activities. But we're aggressively reaching out to communicate to different investors through different venues, which is much more aggressive than before. But one of the reasons is we can do that now with the data. Previously we were selling a technology or marketing a technology, and the technology is fascinating, but someone is going to always say, show me the money, where are the results? And so it's really only until recently that we've had this sort of information in hand to go out and to start exercising that. So, and the other point I would make just on the warrant overhang too is that uh, those warrants, they're short term. Okay, they will expire in less than 20 months now. If they do exercise at $2.05, it represents a significant cash inflow, which is a very inexpensive source of financing going forward, much less expensive than going out and doing an S1 type of arrangement. They're there. It's a fairly simple process. So we either get the money, or if they expire, the overhang goes away. But in either case, I think those are the factors. But what we have to do is continuously increase the breadth of the message, and make sure that we try to keep it as simple as possible. So someone who's not that technically versed can say, "I get it, and this is why this makes sense." And if I buy the stock today, I could double it before those warrants would even convert, and the overhang might even come into play. So I think
That's
that's
great.
best
That's
I can
great.
do.
Last question for me, and then I know there's a bunch of audience questions that I want to get to, but do you guys plan to build a commercial company, or are you going to partner with large pharma? Like, what's the long-term plan here?
The long-term plan realistically has to partner with a bigger company. The commercial infrastructure that's required in order to bring products into the market and commercialize them is incredible. And in our past company, we started as a small development company and then launched two products. And it, 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 the systems and the information that you have to have, hiring the sales force, putting a marketing plan together is easy. It's all the compliance issues that are required. These large companies, they have this down. They've been doing it for years. And so for them, it's easy to take this on. And for them, this is probably a big value creator to add to their portfolio, particularly if they're looking to treat comprehensively skin cancer. This would be a very nice addition to what they have. So the answer is realistically, this is a takeout target at some point in time. And the question is, when is the best time to do that? And the more data, the closer this is to commercialization with the filing with the FDA, the greater the value will be.
Fantastic. All right, I'm going to let Callie get to the audience questions. The floor is yours, Callie.
Great. Thank you so much. If someone has been having Mohs surgery on their face, is this treatment an alternative, or is Mohs a precursor to a tumor that is then treated?
Technically, this would be a precursor to Mohs. Okay. The goal here, based on what we've seen in the results of the study thus far, is that if we cannot eliminate the tumor, the cancer, which would then, if we did that, we would not have to do the Mohs, right? If we do not limit, at least we can what we call debulk it, or to shrink it, if you will. And shrinking it is important because it requires a smaller injector in the term incision into the skin, which will basically put you in much better position. But this would be likely what you would try first. You wouldn't try it second with regard to Mohs.
Okay, great. Thank you so much. And what is the biggest bottleneck that you're working through right now that maybe you didn't cover, or is there one?
Bottleneck. Let me think about that just for a moment. Um, I don't think anything that we've run into at this stage of our development is not something that we hadn't uh, anticipated in terms of the planning. I mentioned earlier to Steve that in the very beginning, the important thing for me was to surround myself with people who have actually had a lot of experience in drug development. And so the key to a very good project plan, which has many, many moving parts, more than you would want to look at on an Excel spreadsheet. You anticipate what these things are going to be. So much of this is all planned through. Sometimes you may find that there's an unexpected hitch somewhere, but maybe a vendor that you wanted to work with, they choose not to work with you. That delayed us a little bit on making registration material about 6, 8 months ago. But we always had plan B, right? Because you always anticipate, you always want to have a fallback. So I don't want to say that this is a cakewalk. It's not, but most of it is addressed through very, very thorough planning. And then if not, then you rely on your experience. It's like the pilot on a plane. Typically, it's a pretty simple thing until you end up having something that's a little bit more of an emergency, and then you got to, adapt to the situation and make it work. So.
Great. Thank you. What changes have you observed in immune cell infiltration or activization within the tumor post-treatment?
Post-treatment. I don't know, Jennifer, you want to handle that since you've been looking at a lot of the detail in a compilation of data in its most minute form. But
Post-treatment.
and are you seeing?
Well, we are looking at Bob the lesion size and the reduction in the lesion size post-treatment, as well as looking to see at day 113 if there's any recurrence of the skin lesion. So those would be the two things that we are looking at. And we are seeing reductions as well as no recurrence at the tumor site or at the lesion site.
Great. Okay, thank you. Can you quantify the degree and duration of PD-1 gene silencing in treated tumors and how tightly that correlates with clinical response?
Right now, I would not want to get ahead of my skis on that because part of that is involved in assessing the pharmacokinetic data in terms of how long
Yes.
the drug is acting to suppress PD-1. What we do know is that the life of INTASYL in the cells is not that long compared to, say, a monoclonal antibody, which is believed to be weeks, if not potentially months. We believe that ours is measured in days. So, but our treatment regimen, which was 1 injection or 4 injections over a 3-week period, that if it seems like the lesion sizes decrease with over the time period until they get to the 5-week observation point. But I can't give you right now a clear answer on that. That would be someone in our organization probably could answer that. But I can't.
Okay, that's okay. Thank you. So what is the typical time to response after dosing, and how does that compare to standard immunotherapy timelines?
Okay, well, if we're talking about, remember, we're talking about 20 of the 22 patients in this study were cutaneous squamous cell stages 1 and 2. One patient was a stage 4 Merkel cell patient in his last stage, as well as one stage 4 melanoma. So it's sometimes a little bit difficult to compare stages 1 and 2 treatment to stages 3 and 4, which is what the monoclonals are treating. But what we have seen is that sometimes by the time of the second injection, the physicians at each time point, they're looking at how the lesion is changing and what the percentage of cancer is remaining, because ultimately that's what the measurement is at the end. What is the pathological level of cancer that's left in the skin? That's the most important thing, because if that's gone, whatever is left visibly is going to disappear anyways in time. Okay, so you're really looking to get rid of the cancer. And that's being measured in this study. How many patients had 100% total clearance of cancer? How many patients had greater than 90% clearance of cancer? And how many patients had greater than 50%? And then the last category was if you had less than 50% clearance, that was deemed to be a non-responder. So, but I think if you look at the monoclonal antibodies, the efficacy rates, I think for Keytruda is somewhere in the range of about 25% as an overall response, and as the total clearance is about 10%. And on the Libtayo, which is the Regeneron product, that one is about 17% total clearance, and I think about 40 to 42% in overall response rate. So ours in the fifth cohort of the study was 85% overall response rate, with 4 out of the 6 patients that responded having total clearance. So that kind of gives you, but a little bit we're mixing apples and oranges here because it's a different state of disease.
Thank you. How are you thinking about expanding cohorts? More patients in current indications versus moving into new tumor types?
Fair question. For a company of our size and based on trying to mitigate risk, or better yet, to increase the chances of success, we have the most information about what we have in the cutaneous squamous cells. And so that will be our target in the next study. We don't really have enough information on the other two. And while it'd be interesting, for example, to maybe do a satellite study on Merkel cell, which is just to see if we would get more partial responses on that, because I think that was really significant given the fatality rates on that disease. We really don't have the funding to do that at this point in time. You know, if something were to come along, it'd be an interesting exercise. But we really need to focus on where we have the most data, because that's the best way that we can define a statistical plan that will give us the best chances of success in the next study.
Thank you. What is the burn rate and how much runway do you have?
Let's talk about the runway first. It's a little bit easier to answer that. Right now, we have cash that's taking us into the second quarter of next year. The burn rate varies a little bit based on certain programs that we're doing and the stages of them. So, you know, the standard part for our what we'll call our administrative expenses are running somewhere in the range of about 4.9 to $5 million. And you can see that if you look at our 10K, you'll see that that's basically what the rate was last year for 12 months. The development side is different because, for example, we're in the process of developing registration material for our active pharmaceutical ingredient. And the timing of when we basically make payments to that vendor based on stage of completion varies. Okay. So with that, maybe the best way to describe this is that we had about $22 million of cash reported in our 10K at the end of the year. And we're taking it out to sometime into the second quarter of next year. So you can figure an average burn that way, but there is a standard deviation on the average based on some of the development activities.
Thank you. And at today's market cap, as you discussed, what specific milestones do you believe would be most meaningful to re-rate the stock?
Perspective, and maybe the investors may feel differently, but I think when the FDA, after we request and get our guidance meeting with them, that they tell us specifically what we have to do in that next study. And that would be the most significant factor that basically takes a lot of the guesswork out of what the investor is going to be looking at. And it also defines exactly what the burn rate will be going forward once that study is started. Because in the end, the FDA is going to tell us how many patients you should be treating, whether, as Linda or as Jennifer had mentioned, we want to do a phase 2B rolling into a pivotal trial. That'll give us a sense of how many patients we need and the time it will take to do that. So that FDA direction is critical. And in the meantime, if we do end up working out any type of a licensing arrangement with another player that would give further credibility to the technology, and I would qualify that by saying we are not looking to license out at this stage any of the rights in the US because the market is just too attractive and it would shortchange the investors and I think the whole program. But there are parts of the world where people might have a strong interest in this. And if we can isolate the geography so that they can do what they want in those areas, as long as they're studying the right thing and doing the right thing with the drug, and we can trust that, we would consider that. So that could be an inflection point as well. Plus, any other interesting feedback that may come from FDA in the process is leading up to that guidance meeting. All of those things I think will be very important, and those will be coming up over the course of the next 3 months or so, 3.5 months.
Great. And 5 years from now, what does Phio look like as a company?
Well, Phio as a company may actually be part of a much larger company in 5 years. We would hope that if everything is going as planned in the development, that the product would be commercialized at that point in time and in the hands of an expert commercialization company. There may be parts of Phio that remain that would be tackling other compounds that are in the program. I mean, the INTASYL program technology is not one drug; it is a platform, and even the PD-1 protein in the PH-762 drug can cover a number of different types of cancers as well. But within the INTASYL, if you change the sequencing of those synthetic fragments of RNA, you can also be looking at silencing a number of other proteins. So it may continue on its own as a development company looking to do with another protein what we're currently doing with the PD-1 protein.
Great. Well, that concludes the questions. And for the audience, if you do have additional questions, please reach out to me. Everyone out there has my email address. I'm happy to facilitate an introduction here. In turn, they will have all of your information as well and will be reaching out. And I'd like to turn it back over to you, Robert and Jennifer, for any final words before we close out.
Just first of all, I'd like to thank everybody who tuned in and taking the time to hear our story, number one. And secondly, hopefully we left you with the feeling that this is actually a pretty serious disease. And I think it's important for everybody to be, as you start to get to a certain age, or if you've had a lot of sun exposure, regardless of your age, to start looking at getting an annual skin check with your dermatologist to make sure that if you have something, that you get it early, because the consequences of letting this go are, you don't want to deal with that. So that's my one advice to you. Get your skin checked. It will definitely be worth the time to spend.
Great. Well, thank you again. Thanks, everyone. And I hope everyone has a great day.